Namaskar. Welcome to the lecture Brassinosteroid mediated stress responses in plant. Myself Dr. Nitika from Department of Botany, Hansraj Mahila Mahavidyalaya, Jalandhar. So first we discuss that what are phytohormones. So phytohormones are the organic substances which are produced by the plant in one part of the plant body and they translocated to the another part of the plant body where they influence the specific physiological functions. They are usually effective in very low concentration and that concentration range from the 10 raised to power minus 4 molar to 10 raised to power minus 12 molar. Now phytohormones play important role in various diverse growth activities as well as to provide resistance to plant against various abiotic and biotic stress responses. The hormones which are involved in plant against different stress, they are auxin, gibberellin, cytokinin, salicylic acid, ethylene, jasmonids and brassinosteroid. In present lecture, we will discuss about the brassinosteroids. So, brassinosteroids are hydroxylated derivatives of the cholesterol which play an essential role in plant growth and development by influencing the various physiological responses in plant. Till now, more than 70 different brassinosteroids have been isolated from the different plants, and out of these, various brassinosteroids are well studied like 28 homobrassinolide, 24 epibrassinolide, brassinolide and castasterone, etc. Now regarding their structure, brassinosteroid, they contain typical steroid nucleus with 4 fused rings. These rings are labeled as ring A, B, this A, B, C and D. This is called as a cholesterol nucleus which has 4 rings, ring A, B, C and D and alkali side chain, which basically provide different structure level to different brassinosteroids. Now come to the history and regarding, you can say, discovery of the brassinosteroids. So in 1970s, an oily extract from the pollen of the Brassica napus was shown to cause extreme elongation the internodes of the pinto bean, and this was discovered by the Mitchell and his co-worker in 1970. And after that, a major screening efforts were carried out at US Department of Agriculture, and later on an active compound was isolated which is named as brassin. Now why it is called as a brassin? Because the first extract which is extracted from this plant, that is your genus Brassica, that's why it is named as a brassin. Now after that, approximately 227 kgs of pollen from Brassica, they were extracted and fractionated with activity test at each step, and finally 4 milligram of crystals were obtained from the 40 kg of the pollen, and which are later on identified as brassinolide by Groove and his co-worker in 1979. And this brassinolide is basically designated as a first plant steroidal hormones. Now come to the distribution of brassinosteroids. So brassinosteroids are distributed in approximately all the plant kingdom as they are ubiquitous in their distribution. So far they have been found in dicots, monocots, gymnosperm, fern and even alga and bryophytes. Regarding the plant parts in which they are present, the major amount of these hormones is present in the pollens and immature seed. But in addition to this, they are also found to be present in the shoot, leaf, pollens or other plant parts like in their thallus in case of this pteridophyte, etc. Now next come to the physiological effects of brassinosteroids in the plant. So at whole plant level, they basically promote the growth, increase the success of fertilization, shorten the period of vegetative growth, increase crop yield, and in addition to this, they also provide the resistance to plant against various abiotic and biotic stresses. At cellular level, they stimulate the elongation and fission, and they also activate the protein and nucleic acid synthesis, and they also alter the activities of various antioxidative enzymes, and also enhance the photosynthetic capacity and translocation of the photosynthetic product to different parts of the plant. Now next is the role of brassinosteroid in stress management. As we have discussed in previous slide that they play important role in various physiological activity, but in addition to this they provide the resistance to various abiotic and biotic stresses like water stress, thermal stress, salt stress, heavy metal stress, pathogen or pesticide stress, etc. Now first of all we now discuss that what is stress. So stress is any external condition that adversely affect the plant growth, development or productivity of the plant. Now stress can be abiotic or biotic. In present study we will discuss about the abiotic stresses. So abiotic stresses which are arising from an excess or defect in any physiological or chemical environment like water stress, high or low temperature stress that we call as a thermal stress, excessive soil salinity or any chemical stress which may arise from the excessive use of the pesticides rich in heavy metals or air pollutant or any type of the radiations. Next is the response of the plant to environmental stresses, that how plants they respond to the different environmental stresses. So this is depend upon the first on the stress characteristics like their severity, duration of the stress, number of exposure or the combination of the different stress. And secondly depend upon the characteristics of plant, that which particular organ or tissue of plant which is exposed to the stress, stage of the development in which that particular plant is exposed to the stress or genotype of the plant. Now depending upon if the plant is able to show the resistance to that particular stress, it may lead to the survival or the growth of that plant, or if it is susceptible then it may lead to the death of the plant. Now there are different environmental factors which induce the production of the reactive oxygen species in the plant cell. So from the various studies it was evaluated that any type of the stress ultimately induce the oxidative stress by enhancing the production of the reactive oxygen species. Now heavy metal stress is basically induced by the excessive use of the heavy metals in field, or it may be because of the, you can say, excessive use of the fertilizer, or it may be leaching of these heavy metal from any type of the rocks, etc. So heavy metals are those which have the density more than 5 gram per cubic centimeter like zinc, chromium, nickel, copper, etc. Usually they are essential as a micronutrient for plant, but they are toxic at the higher concentration. So heavy metals also inhibit the growth or development of the plant, and it also induces the oxidative stress by enhancing the production of reactive oxygen species like superoxide radical, hydrogen peroxide, alkoxy radical or hydroxyl radical. These reactive oxygen species basically initiate the lipid peroxidation of cell membrane, or it may degrade the proteins, lipids or nucleic acid. Now heavy metal induced oxidative stress also induce the other cellular damage in the plant, like it may damage the protein, DNA or lipids, and also it induce the membrane peroxidation and may lead to the depletion of the antioxidant like glutathione and NADPH, which may sometimes lead to the cell death. Now to combat this situation, the plant cell may activate the certain antioxidative enzymes or certain antioxidative substances which ultimately constitute the antioxidative defense system. Now next abiotic stress is the salt stress, which is also one of the major abiotic stress to the plant after heavy metal stress. It also inhibits the growth of the plant by inducing the physiological drought where plants are not able to absorb the water from the soil. So as salts, this heavy metal stress, the salt stress also induce the excessive production of the reactive oxygen species which cause the oxidative stress in the plant cell. So this is the pictorial presentation of the different plant responses to the salinity stress. Like there is enhancement in the level of the certain compatible solutes or osmolytes like proline or other types of sugar, etc. Enhanced level of the polyamines were observed in the saline stress plants. In addition to this, it also induce the production of the stress related proteins like LEA protein, that is late abrogenesis proteins, and chaperones or heat shock protein, and it also alter the activities of certain antioxidant and antioxidative enzymes. Now next come to the point Asada Helival pathway. So basically when any plant is exposed to the any type of the stress, the oxidative stress which is produced in response to that particular abiotic or biotic stress result in the excessive production of these reactive oxygen species like superoxide radical, hydroxyl radical, or alkoxic radical, or hydrogen peroxide. Now these reactive oxygen species are need to be removed from that environment of the cell, and that can be possible with the help of the activity of the antioxidative enzymes and antioxidant. These army of the antioxidant and antioxidative enzyme act in a coordinated manner which constitute a Asada-Helival pathway. So Asada-Helival pathway basically consists of the different antioxidative enzymes like SORT, that is superoxide dismutase, POD, guaiacol peroxidase, epox, ascorbate peroxidase, GR, glutathione reductase, DHAR, that is dehydroascorbate reductase, MADHAR, that is monodehydroascorbate reductase. So these all are acted in a coordinated manner. Now out of these antioxidative enzymes, this SORT, that is your superoxide dismutase, act as a first line of defense which dismutate the superoxide radical into hydrogen peroxide, H2O2. Now this H2O2 is also one of the reactive oxygen species and it is toxic to the plant cell, so it has to be removed from the plant cell with the help of the different other antioxidative enzymes. So this H2O2 can be removed from the plant cell either with the help of the enzyme catalase which change this H2O2 to water plus half oxygen, or it may be reduced with the help of the enzyme ascorbate peroxidase which use the ascorbic acid as a reducing substance and change this H2O2 to water. Now after this ascorbic acid is changes into the monodehydroascorbate. Now this monodehydroascorbate may be spontaneously changed back into the ascorbate to refill the pool of this reducing substance, or it may be changes to the ascorbate with the help of the another enzyme, that is your monodehydroascorbate reductase which use the NADPH as a reducing substance. Secondly, this monodehydroascorbate radical can be spontaneously changes into the dehydroascorbate. Now this dehydroascorbate is again need to be changed into the ascorbic acid, and that is possible with the help of the enzyme dehydroascorbate reductase, that is your DHAR. Now this dehydroascorbate reductase utilize the reduced glutathione as a reducing agent, and this reduced glutathione during this reaction is changes into the oxidized form of the glutathione. Now this oxidized form of the glutathione is again need to be changes into reduced form to refill the source of these reducing substances. So that is possible with the help of the enzyme glutathione reductase which use the NADPH as a reducing substance. So in this whole process these enzymes they are acted in a coordinated manner. Now in addition to this there is involvement of the certain antioxidants like vitamin E and carotenoid also which help to change this reactive oxygen species into non-reactive oxygen species like water, etc. Now come to the role of these brassinosteroids to overcome the heavy metal and salinity stress. So as we have discussed earlier that this any type of the stress it induce the production of the excessive reactive oxygen species. Now these excessive reactive oxygen species are get reduced to the non-reactive oxygen species with the help of the antioxidative enzymes and antioxidant. So these enzymes are basically activated by the Brassinosteroids. So you can say their activity is further stimulated with the help of the brassinosteroid, which help to overcome the oxidative stress which is induced by the heavy metal and salinity stress. So next is the possible mode of action of brassinosteroid under stress condition in case of the plant cell. So brassinosteroids, they are basically acted via receptor ligand complex that bind to the nuclear or cytoplasmic site to regulate the expression of the specific stress related gene. So brassinosteroids are perceived by the extracellular domain of plasma membrane localized receptor, that is your BRI1. Here BRI1 is brassinosteroid insensitive leucine rich repeat receptor kinase. After the binding of the brassinosteroid with this BRI1, a second protein or co-receptor, that is BAK1, which is labeled as BRI1 associated receptor kinase, heterodimerize with BRI1, which in turn induce the autophosphorylation of BRI1 and BAK1. And this in turn activate the cascade of signal transduction reaction within the cell. Now after the phosphorylation of these receptor, it lead to the localization of the brassinosteroid responsive protein, which include BZR1 and BES1 inside the nucleus by inhibiting the activity of the BIN2, which otherwise inhibit the localization of these brassinosteroid responsive protein, that is BZR1 and BES1 inside the nucleus. After translocation of these BZR1 and BES1 protein inside the nucleus, they activate the certain brassinosteroid responsive gene expression, which ultimately lead to the synthesis of the various metabolite and stress protective proteins, as well as certain compatible solutes and proteins which are synthesized by brassinosteroid responsive gene mainly include the LIA proteins, that is late embryogenesis proteins, or the chaperones like heat shock protein, etc. In addition to this brassinosteroid receptors, which include BAK1 and BRI1, their phosphorylation also stimulate the activity of the vacuole localized NHX, that is your sodium ion hydrogen ion antiporter, and VATPase pump, which in turn move the excess of the heavy metals ions and sodium ions to the vacuole. So in this way, this whole receptor mediated process protect the plant against the heavy metal induced or salt stress induced stress and provide tolerance to the plant against this type of abiotic stresses. So brassinosteroid have potential to act as a stress protectant when it is applied at appropriate dose and at correct stage of the plant development by shifting the certain biochemical processes or activating the certain antioxidative enzymes and antioxidant, etc. So these stress protective properties of brassinosteroids are promising from practical point of view, and they provide new insight for plant protection based on the employment of very small amount of these eco-friendly natural substances as compared to the application of these highly toxic other chemical substances. So that's all for brassinosteroids and their role in the stress protection in plants. Thank you.